Oh, that was aggressive. All right. Good day, everyone. Thank you for coming to this talk. I will hand it over to Rachel to talk about the ManyBirds study.
Wonderful. Thank you. Of course, I just had a knock at the door. Just gonna ignore that. Yeah, it's really lovely to be here, and I'm particularly excited today to be sharing for the first time our results of ManyBirds Study 1 on the evolutionary drivers of neophobia across the avian clade. Um, and I'm presenting this on behalf of our ManyBirds project and all our collaborators and our Study 1 leadership team, who I've listed here. Awesome. So I think, as we are all aware, there are many different fields that have been affected by the reproducibility crisis, including the field of comparative cognition. And this has helped us to identify that at least some of the field faces issues in terms of methodological constraints, small sample sizes, few species representation. And indeed, in a review that my colleagues and I completed a couple of years ago, we reviewed avian cognition and behavior research across a 5-year period until 2020. And we identified that only around 1% of potential species were represented across 4 bird orders, and that these were coming very much so from lab sites as opposed to representation within field sites and zoo sites. And that they were largely also coming from 4 countries: the UK, USA, Canada, and Austria. So as you can imagine, this raises concerns in terms of potential limitations of the field as it stands and our results so far, and limits how accessible and inclusive and representative our research may be, and how generalizable and replicable it may be. And in general, just I think much wider than comparative cognition, there are aspects raised in terms of concerns around research transparency, which we can rectify with open science procedures as much as possible. But for me, certainly, it's these concerns about, really, are we capturing the variation within the animal kingdom? So you might think, why are we interested in birds? So one of the biggest advantages is the sheer size and diversity of the avian clade. It's over 10,000 species. They occupy many different niches. They face similar social ecological pressures to mammals. There's been a case raised for convergent evolution of intelligence, for example, corvids and parrots compared to great apes, and even with children. And lots of comparative work around looking at the study of the brain architecture and function in birds and other taxa in order to explore cognitive evolution. So my colleague, Dr. Megan Lambert, and I were inspired by lots of existing big team science projects, like Many Primates, Many Babies, to found the ManyBirds project in February 2021, a wee while ago now. And we now have core and Study 1 teams, and these are largely early career researcher driven, which is one of the things that I also really like about the project. And one of our major aims, and certainly for me personally, was to try to help change a narrative from science only being highly competitive to, and very closed in some cases, to being much more collaborative and really coming together to work together to pool our resources in order to address some of these big questions, like the broad evolutionary drivers of cognitive and other behavioral abilities. And also to incorporate these open science practices within our project to study avian cognition and behavior. And I'd say we've made some good progress. We have 3 publications under the project. We are involved in large funding bids with some of the other big team science projects. And you can find out more about all of this through our website or through our Twitter account. So one of the studies we were doing alongside this review that I mentioned, and also launching the ManyBirds project, was to almost as a pilot study, if you like, to look at the social ecological correlates of neophobia in corvids. So these are members of the crow family. So neophobia is essentially an aversive response to novelty. It can be novel objects, food, environments, predators. There's a huge variety there. And we're interested in neophobia because it allows animals to navigate risk, to interact safely with their environments or as safely as possible with their environments, and therefore has really important effects for adaptability and survival. So in this study, we tested object and food neophobia in 10 corvid species across 10 labs wide with over 200 subjects. And this is just the results for the object neophobia. We did identify some social ecological drivers, for example, relating to habitat, to sociality, and to foraging behavior. So a dietary related category to do with caching. And we did find some of these differences. So just for one example, that those corvid species that use urban habitats as well as other habitats typically show a lower neophobic response. Oh, I think somebody's not muted. But one of the big kind of questions that certainly I was taking away from the study and other colleagues was, are these findings corvid specific? Corvids are in some ways quite an unusual group of birds. Or are they really more applicable are these findings more applicable across birds more widely? So across other orders and families, etc. So we decided to select neophobia as the focus of the ManyBirds Study 1. We had a pre-registered protocol for focusing on object neophobia. We wanted to keep it really simple in order to facilitate standardization across labs, field, and zoo settings. We had an open call for over a year for data collection that closed beginning of around April last year. And we had three major objectives. We wanted to be able to compare neophobia across different orders and species and predicted that we may find some differences. We wanted to specifically then test how these differences may be driven in terms of social, ecological, and other factors. And we made hypothesis-driven predictions around diet, sociality, habitat, range, and also domestication. And I'll just mention the domestication was one factor that we added post hoc because of the final sample, including several domesticated species. I won't go through all these predictions in detail, but you can get an idea here. But one that I will highlight is that we did have an expectation that generalists, both in terms of diet and habitat, would typically be less neophobic than species that were more specialist. So relating to the need to be able to essentially explore and utilize different habitats, different resources for those generalists. And we also wanted to look at responses on an individual level and predicted that we would find some repeatability across individuals in how they respond, both across time and also different contexts. And again, we expected that we would find individual repeatability primarily because we were using quite short delays between test rounds, around 2 weeks. And we were only focusing on object neophobia. And also that this was supported by lots of different previous studies, usually with single species or a small number of species. So we were really happy to have a load of interest in joining this study. And we were able to test or to collect new data in 136 species across 25 orders with over 1,500 subjects. And this was only possible through the collaboration of over 100 collaborators at 50 institutions across 20 countries. So we've gone way beyond those 4 countries that were initially identified in the review I mentioned, and across 6 different continents. So I think that's really cool in itself. as I say, we took quite an inclusive approach to including data from both labs, zoos, and the field. And just to give you a little snapshot, you can see quite a range in the variation of species that we were able to test. So from your passerines to your parrots to your hornbills to your storks, all the way kind of at the top of this figure to your paleognates, so those are your emus, cassowaries, etc. So there's quite a lot of variability there. So as I say, we use this quite simple standardized protocol where we tested responses to novelty, and our main measure was the latency to touch familiar food in the presence or absence of a novel object. And we ensured that there was a consistency with those objects in terms of things like their colors. They were all visual. And the size was varied in terms of the species. So this one here is the cassowary, for example, down to this one down here, which is the common waxbill. I hope this video is going to show, I won't maybe play the whole thing, but I just wanted to give you, I think it's really nice just to see the different setup. So this is obviously a wild setting. This is the redback shrikes. Just to give you an idea of how some of the setups looked. So we did ask all collaborators to send us pilot videos of just the familiar food and also photos of their objects before any data collection was completed. So this is a caracara, again, a wild setting. Caracara, as you see, immediately going to touch the object rather than even the food. They're very well known for being a very neophilic species, so almost attracted to novelty. And now going for the food. Again, a zoo kind of setup. Here we've got our lovely emus. So this is our object approaching near the food. We've sped it up a bit because they really took quite a long time. Again, this quite a neophobic response here from the cassowary. Again, here's our object and our food. Really taking a bit of time to come and interact. Andean condors. Quite a cautious approach, if I can say so. And again, kind of lab settings. So here, for example, the green macaws. This is a control condition. And the novel object condition. So again, keeping everything consistent as much as possible within subject and species. Archaeas, again, very neophilic species. And I'll just jump forward now so that I don't lose too much time. And so for our three main objectives, we did find these order level differences. You can see the results here from the control, from the test, so that's the novel object data. And then also kind of crucially, we use these different scores, which are a compiled score of the control and the novel test condition. And so it helps control for any potential site differences. And what we find here, again, as I say, there's this variation. So for example, the grebes and the cranes showed these kind of highest neophobic responses, the biggest difference between the control and test latency. And the woodpeckers, falcons, and pheasants showing the lowest neophobic response here. So that's our galliforms down here, our pheasants and allies. And so you might kind of have that as a result, and you might be able to speculate from that, that there may be different social ecological drivers within some of these orders. But what the really nice thing is with the data set that we have is that we can actually test for this. So what we did is tested these several social ecological drivers. This is the results using the different scores. As I mentioned, those are the novel object minus the control latency data for each individual. A higher score equals more neophobic. And what we find is these clear kind of consistent results in terms of dietary breadth. So that's that difference really between feeding specialists being more neophobic than feeding generalists. And migration status or migration pattern, where again, we find that these migratory species are actually showing a higher neophobic response than the non-migratory species. if I didn't mention already, I will say that this is all in prep. It's due to be submitted next month. So you'll be able to see the preprint at that point. When we look just at the raw data, which allows us to check for consistency in some of those major findings, where we also look within the control and the test data, but within each condition, we find again that consistent finding of migratory pattern and also in terms of dietary breadth. And interestingly, we also have a couple of other factors that pop up, one of which is the social bond effect, where solitary species are more neophobic than long and short-term species. But I have some slight reservations around this particular factor because there was only one species, the greater rhea, that was solitary. So I would treat this one with caution. But we do find this domestication status effect where non-domesticated species were more neophobic, which is in line with our prediction. And because of that domestication effect, we did look at our full data set and remove 7 domesticated species in order to assess those non-domesticated species only. And when we look at this reduced data set, again with the raw data, interestingly, the migratory patterns, the dietary breadth results, again, they hold, but we also now find that there are effects of habitat. So 2 of our habitat factors jump out. One is the number of habitats. So that's again looking at the kind of specialist versus generalist category. So specialists, again, more neophobic than generalists. So that's a consistent finding. And interesting, habitat diversity. And this is where we find something that we didn't necessarily predict, that species from more complex habitats were more neophobic than those from simpler habitats. And I'll explain a little bit more about that finding in the discussion. We did find individuals to be largely repeatable, both across time, so between these test rounds. So we tested at least 1 or 2 rounds, in some cases 3, and contextually repeatable between the control and the novel objects condition. So in summary, we found, yes, we support our findings of some similarities and differences in order comparison. We identified some social ecological drivers, particularly in terms of diet and range, I would say are the most consistent responses there. And these slight differences in terms of what we predicted in terms of no effective territoriality. There was an effect of social bonding, but as I say, treating that with caution. But also, in some ways, surprisingly, this differing pattern of data with the complexity of the habitat, and I'll discuss that in a moment. And again, we did find this individual level repeatability, which is helpful as it indicates that this is a stable trait. So, as I say, one of the things that we really have to keep in mind when we're testing across so many different sites and species is that there are some unavoidable site differences. And one of the major controls that we use for this is those different scores that I've mentioned a few times. And I think that's where it's really important to have those control trials. We did find these drivers, particularly in terms of diet and range, and that these also are largely in line with the predictions that not only we made, but also in terms of 2 major hypotheses for neophobia, in terms of the neophobia threshold hypothesis, which makes this prediction that generalists will be less neophobic than specialists. So we see that with both the dietary breadth and the habitat use findings. But interestingly, we also find evidence for a non-mutually exclusive hypothesis, the dangerous niche hypothesis, which relates to the level of danger or risk in the environment. And that's where these habitat diversity and range variables seem to support. And as I say, that individual repeatability is important, I think, to test for, and useful that to have that finding that there is that consistency in individual level responses, suggesting this is a stable behavioral trait across the avian clade and likely also in other taxa. So of course, there are some limitations that we acknowledge and that some of which are outside of our control, but we have tried to account for in our analysis, for example. Standardization across these different sites and across many different researchers is challenging, as I know a lot of big team science researchers are also aware of. Um, and as I say, some of the things that we've done in terms of needing those pilot videos, the novel object photos to be submitted to us beforehand, a very simple pre-registered protocol, all important things. We also publish all the study site and subject details, and within the analysis we control for this, for example, with those different scores, also including the factors like domestication. We note that because we were inclusive in terms of lab, field, and zoo settings, there are some potential differences there between captive and wild individuals. But we didn't have enough subjects from field settings to be able to test that within the analysis, but that would certainly be something worth looking at in future. And of course, we also recognize that there's a lot of variability within those individuals that, again, we cannot control for in terms of their housing, their rearing, their previous experience, etc. And I think the main thing for us was just to try to include as many subjects and species as possible. So we were very open to that. And as I say, try to control for that in analysis and be transparent in those details. So lots of future research, things that we're excited about in terms of potentially looking at other types of neophobia, like food or predators. Of course, other drivers in terms of things that are more developmental or proximate. Looking at the relation between neophobia and other cognitive or behavioral abilities, I think will be really interesting. For example, things like innovation, problem solving. I think this neophobia data and this approach really does have important implications for comparative cognition in terms of understanding the drivers of neophobia, but also, you know, for example, in terms of what we've found in species that are less represented or threatened, and you know, some of those zoo species. This allows for us to learn a bit more about those species that may be useful from an applied conservation perspective, you know, using kind of how animals respond to novelty to inform conservation actions like selection of individuals for reintroduction. And again, that's where it's important to know that this is a stable trait and not necessarily a kind of flexible trait that we can't use. So lots of challenges and lessons, as we all, I'm sure, are aware of. Some of the ones that certainly sprung to my mind as a project lead on this were that a lot of our lead team in particular, in fact, all of our lead team, do this as a, you know, voluntary roles alongside our own jobs. We're not often allocated time or money to do it. It can have quite a high workload. An awful lot of big decisions that need to be made that affect the project, affect collaborators, etc. Can be challenging. Learning a lot of skills around project management, just the sheer length of this study. We've been working on this study, as I say, since 2022. It's been a good couple of years to get this through to the point of almost submitting. But lots of really positive things that have come out of it in terms of really just feeling like we're trying to make a difference here in how science is done, trying to make it more inclusive, trying to be more supportive of each other, more collaborative in how we do things. And we'll certainly be reflecting on a lot of the lessons that we want to learn from what we've run, what we experienced in running this study moving forward. And the next really exciting steps once we submit this manuscript will that we will have an open call for taking a consensus-based approach to selecting the next studies for ManyBirds. Some of our collaborators have already been making some really great suggestions, and so we're starting to look into those options. For example, looking at avian communication, which may take a more targeted species selection, looking at factors like play, exploration, innovation, a more foraging focus, for example. We may be joining some other big team science projects such as the Many one on reversal learning. We'll certainly be putting that out to our collaborators. But in general, we'll be trying to ensure that our studies are kind of really possible and in line with what we aim for within the ManyBirds framework, as I say, in terms of that inclusivity, accessibility, representation. And so, of course, we want studies to be possible to test multi-site, multi-species, and ideally, from a data collection perspective at least, low cost time and labour. I don't know if those things necessarily apply to the lead teams, but hopefully that'll be something we will review going forwards as well. So just that's me. A big thank you to particularly the Study One leadership team who have just been so wonderful to work with. Just a joy to have any of our meetings, always makes me happy and smile. And to all of our ManyBirds project collaborators, it's incredibly refreshing to see that there are so many people within these fields that we work in that are so open to really taking this approach, these kind of approaches together. It's wonderful. And do look out for our upcoming proposal call for anybody interested. We're always taking on new collaborators, new species, new sites. So lots of opportunities there. Keep an eye on our website and feel free to get in touch if you have any other questions. Thank you so much.
Great talk. We have a couple more minutes if anybody has any questions now. Otherwise, we will just hang out. Yes, Drew.
Hey, Drew.
Hello. Yeah, it's so great to see this first public presentation of these results. Yeah, this is really exciting. I'm really glad I get to see this. Where to start with questions? I guess first question. I was curious if you had considered or ran into issues or if there was any sort of story behind looking at the results in like a phylogenetic perspective.
Yes, so we did control for, sorry, I forgot to mention that. We did control for phylogeny for the objective two, so looking at the social ecological factors. And that's also interesting because we weren't able to do that for the COVID study with having too few species for that. And we did look at the effect of phylogeny in that first objective as well with the order differences.
Okay, so everything is already like controlled with phylogeny. Okay, great. Yeah, okay, that's really interesting. Um, I could ask another question, but I don't want to hog the floor.
It's fine for me.
Okay, I'll ask one more. So obviously, yeah, there's so many, these birds are very different, very different setups. And I was kind of wondering how you navigated the site differences in terms of like, how did you try and control the differences to make them as minimal as possible while still letting the birds do their own thing? Because like one of the things that struck me from one of the videos was it looks like it quite differs how close one of the bowls is versus the neophobic object is, depending on how the bird approaches it and so on and so forth. And I know there's all kinds of sources of variability, but I was just sort of curious what sort of approach you took to that.
Yeah, it's certainly a major challenge, and I know you know this from many primates as well. Um, yeah, so I mean, I think the pre-registration certainly outlined a lot of these issues. So that was around the actual object construction and the size of the object, the distance from the familiar food it was adapted slightly for the size of the species. So some of our larger species, there'll be a bigger gap there. In terms of the differences in the sizes of enclosure, etc., we have no control over that. The other thing we have no control over is whether species were tested individually as they were in the COVID study previously, because that was all lab birds, or in a social setting, or even with other species present. So those are things that there are kind of unavoidable differences with. But I think that this is where the control trials and those different scores are really crucial, because that at least allows us that on an individual level to ensure that whether or not that object is present, this is how, quickly your subject would typically respond, typically approach the food. without the object being present there. And I think without that, I would be even more concerned about the site differences. And one of the things I was debating with Vedrana, who's one of the study leadership team the other day, was, can we even present these raw data results? Should we only be focusing on the difference scores? But we decided that from a place of open science kind of transparency, we do want to show both of those approaches. And what is, for me, certainly what's good to see is that we do see consistency in the two major factors across the different approaches, regardless of whether we chuck out the domesticated species or not. So I think we're just trying to take a really kind of transparent approach with that analysis as well in the paper.
Okay, yeah, super interesting. Thanks again.
All right, so we are at time, so I will let people get to the next session if you're going to that. Thank you again for presenting. This was really interesting. Yeah, have a great day, guys.
Great. Thank you so much, everyone. Lovely to see you.